Thanks. Let's go to question number 10. Question number 10. If the menstruation occurred to a woman in the first day of the month and she wanted to use contraceptive pills, which day of this month this woman could begin taking the pills? What are the contraceptive pills make? They are consist of synthetic hormones, progesterone and estrogen. They stop the menstrual cycle, but it has a day. Which day we must choose to start these pills? The choices: A, 1, B, 5th day, C, 7th day, D, 14th day. Which day? Which day is suitable for the hormones pills? Let's go to the answer. It must be the 5th days. The 5th days to start to take the hormones will stop the evolution. Okay, let's go. Yes, reproductive again. I like reproductive. Reproductive is the process to increase the number of offspring to ensure continuity of the living organism or the species. Let's go to this question, 11. What is the difference between the two flowers, A and B? A and B. This flower is hermaphrodite. Hermaphrodite means it contains the female organs and male organs. They are found together in the same flower. Let's read the choices. Number A, types of pollination. Are they different from the type of pollination? What is the meaning of pollination? Pollination is a transfer of pollen grain from andrecium to gynecium. From the male gamete transform from their organs to the organs of female to make fertilization. That's pollination, the process of transfer the pollen grain from two. Let's go. Number A, type of pollination. Yes. B, sex of the flower. No, they have the same sex. Hermaphrodite contains the two organs, male and female. C, number of pollen sacs. Number of pollen sacs doesn't appear here. They are maybe 4, maybe less than or more than, but ideal they 4. D, number of ovules. Number of ovules, no. The number of ovules here 4 in the scene. So what are the difference between the A and B? It must be the andrecium. Okay, type of pollination. Type of pollination here depend on the first one, not a self-pollination, but the second one, A, is a self-pollination flower because the stigma is shorter than the antheridium. Yes. Let's go to another one. It is the stigma is longer than the anther. So the answer is types of pollination means how to transfer the pollen grain from anther to the stigma. Here, number A, the type of pollination is a self-pollination because anther transfer the pollen grain to the stigma by air. The second one, the stigma is higher than the anther, so it will not be self-pollination. It is a cross-pollination, or the pollen grain comes from the other flowers. Let's go to 12. 12. Which of the following best describe fallopian tube in the normal woman? Where are the fallopian tube? Fallopian tube is a part of female reproductive system. Female reproductive system consists of 2 ovary, 2 fallopian tube, 2 fallopian funnel, and other parts Where are the fallopian tube found? Inside the abdomen of the female. What are the function of fallopian tube? Let's read the choice, then we make another decision. A, which of the following best describe for fallopian tube? I will describe the fallopian tube. Number A, cilia of the tube move toward the ovary. No, it cannot move toward the ovary. It moves away from the ovary to collect the egg or the ovule to become inside the fallopian tube. B, end of the tube is wider than the beginning. End of the tube? No. End of the tube, it will become inside the uterus. It will not become wider. C, beginning of the tube is attached to the ovary. Beginning? No. Beginning at the ovary, not attached. D, cilia of the tube move toward the uterus. Yes, the cilia is attract the ovum to go inside the uterus. Let's check the answer. Yes, the answer is cilia of the tube move toward the uterus. Yes, move toward the uterus and push the ovum to go inside the uterus. 13. Which of the following stages of human embryonic development has the highest rate of DNA duplication in the embryo cells? When the DNA become duplicate. The DNA become duplicate at the division, at the cell division. I need more cell division to grow the embryo. Which part of this will need more growing, more mitosis, more cell division? Let's see. A or B or C or D, it will be delivered. A, it's not a more active division, but B, it's more active division, more division to more growth. Let's go to C. That means B is more duplication DNA to make more growth, to make more division. Let's go to 14. 14 is a question speak about which of the following amnion method precedes the other one in its occurrence. Which one of them preceding the other? There are 4 choices. The first choice speak about increasing the number of receptor formation of the cell wall. Which of them is preceding the other? Formation of the cell wall is a barrier. One is the first one in the formed of the cell wall. Cell wall formed before anyone. Number B, deposition of gum. Thickening of the stem epidermis by keratin. Which of them is preceding the other? The first one is deposition of gum. Deposition of gum happens when there are infection in the cell wall. So it's not preceding the thickening of the stem epidermis by keratin. The thickening of stem epidermis by keratin is come first. C, thickening the cell wall by lignin, and the other one is producing antimicrobial protein. Which of them is come first? Thickening of the cell wall by lignin is come first, not by producing antimicrobial protein. The antimicrobial protein is come the second one. Number D, producing detoxifying enzyme swelling the cell wall. Which of them is come first? The swelling of the cell wall is come first. So the answer must be C. C is thickening the cell wall by lignin is come first because it's happened before producing antimicrobial proteins. Now we will study the figure. Studying the figure is very important in answer the question. You must study the figure to make a decision about the answer. Let's see this figure, lymphocytes. Lymphocytes, there are 2 lymphocytes. Binding 1 infected cell. Binding 2 lymphocyte. Cell function stop. So one of them has a function. What? Dies. Let's go. The figure, let's read the question to make the decision when what of them is the answer. The figure illustrates the role of 2 types of lymphocyte. Study it and answer. What substances are produced from 1, 2, respectively? Let's go to the choices. The first one, complement, interleukins, lymphatic, toxins, lymphokines. C, interleukins, complements. D, porphyrin, cytokinase. Which of them is the choice? Let's go. The choice is lymphatic toxin, lymphokinase. Yes, that means this phase of immune must be formed lymphatic toxins, lymphokinase. Let's go to 60. What is the result of removing the spleen? Oh, spleen is an immune organ. Spleen is very important in blood formation and blood cell die. Anything die inside the cell, inside the body, it will make recycling. It will use it again. Let's go to the question and the choices. Number 8, decreasing the number of memory cells in the blood. Is the spleen make the memory cells? B, increasing the number of senescent red blood cell. Senescent means aged or at the end of age. Inability to produce antibodies. D, inability of thymus gland to differentiate the lymphocytes. Which of them is the choices? Let's go to answer. To answer, increase the number of senescent red blood cell. The red blood cell will go to the spleen to recycle it. But here, when we remove the spleen, many number of these red blood cells are sent. Go to aged, will become die, and not to remove to recycling again. 17, when doing a blood analysis for a person. Blood analysis means how are the quality and the quantity of the level of substances inside the blood. A pieces of bacteria was found in the blood sample. Wow, it infected by a bacteria inside the blood. Let's go to see which immune cells are responsible for protecting this person. When the bacteria found inside the blood, it must have a type of cell to protect this person. Let's read the choices. Number 1, A, natural killer cells. B, plasma B cells. C, memory T cells. Cytotoxic T cell. Let's go to the answer. The plasma B cells are found in the blood. The plasma B cells protect this person from the bacterial infection inside the blood. 18. Hemoglobin protein is produced by mature red blood cells, but liver cells from the same species do not produce hemoglobin protein, and why? Now we find the reason why the hemoglobin protein is not formed in the same species. Let's read the choices to make a decision. Number A, the gene for hemoglobin is missing in the liver cells. The gene must be found in the DNA and form a new type of protein. The protein here is hemoglobin. It must be found, not blocked, not produced. Number B, the gene for hemoglobin is turned off in the liver cell. Means it will stop by any disease or anything, maybe or not. Let's go to C. The gene of hemoglobin has mutated in the liver cell. Mutation. Mutation is the change on DNA by any way. Let's go. D. The gene for hemoglobin is activated in the liver cell. Activated is not suitable for don't produce hemoglobin protein. It's not suitable for that. Activated means increase the number of hemoglobin and the production of this hemoglobin. So the answer will be C. The gene of hemoglobin has mutated in liver cell. Mutated means stop for a reason. Stop for a reason. Any reason. Maybe exposed to some rays or anything like that. Let's go to 19. A DNA molecule in which both strands have radioactive thymine is permitted to replicate in an environment that contains non-radioactive thymine. What is the right number of DNA molecules which possess some radioactive thymine post 3 duplication? 3 duplication? Let's read the choices, then we will decide. One such molecule, no, they are 3 replication. 2 or B, 2 such molecule, 2, maybe we follow that. C, 4 such molecules, 4, it's more than 3. D, 8 such a molecule, 8, it's too much. Let's go to the answer. The answer is 2 such molecule. So we need to read the question again to predict that the 2 such molecule, yes or no. There are non-radioactive thymine. What is the right number of DNA molecules which possess some radioactive thymine post 3 duplication? Yes, there are 2 such molecules. That's right. Let's go to 20. 20, speak about amino acid and protein. Let's go to read. Each amino acid in a protein is specified by amino acid of the protein. It is the building unit of it. Several gene, promoter, and messenger RNA molecule, a codon. Wow. Protein, amino acid, we need a code. Yes. Let's go. A codon. Codon. It is needed for protein specified.